Greetings! My name is Amanda Anqueira. I am a graduating senior at the University of Puerto Rico at Rio Piedras, and today I will be talking to you about my research project, which I have been doing under the mentorship of Dr. Alfredo Getzi, titled The Molecular Mechanisms of Ethanol Neuroadaptations. In this lab, we aim to understand the effects of alcohol consumption and abuse, or alcoholism, in the brain using Drosophila melanogaster as a model organism. Alcoholism is best described as a disease in which a person is addicted to ethanol and the capacity to stop or control consumption is affected. This physiological dependence is nothing more than an adaptation to the chronic consumption of alcohol, which also involves withdrawal symptoms. A study published by Robinson et al. Evaluates this concept of neuroadaptation and its effect on learning. In the study, larvae were grown in two different groups. One group was grown in food lacking ethanol, and the other group was grown in food containing ethanol. At the end of 6 days, both groups are transferred to fresh food with and without ethanol for an additional 6 hours, resulting in 4 groups. At the end of the 6 hours, all the larvae were trained in a heat shock olfactory conditioning assay, in which an attractive odor is paired with a thermal shock. After the training, learning is measured with a test where larvae must choose between the odor paired with a shock or a vehicle odor. A higher learning index meant the larvae had successfully avoided the odor paired with a shock and learned successfully. The results from this study showed that control and chronic ethanol larvae had a similar learning index. Thus, they had learned essentially the same. When focusing on acute ethanol and withdrawal, they both had low learning indexes. Thus, they had not learned. So we clearly see how the acute exposure of alcohol results in a deficiency in the learning index, while the chronic exposure does not present this deficiency. This is a clear indicator that the constant presence of alcohol induces a series of neuroadaptations that mitigate the effects of alcohol on learning. 3 questions arise from this study. The first one, how does gene expression change in these chronically exposed flies to alcohol? That is, which genes may be contributing to these neuroadaptations? The second question is, what other behaviors change as a result of these neuroadaptations? And third, how does neuronal and synaptic morphology change? To study the neuroadaptations to alcohol, 2 groups of larvae were grown. One group was grown in food that was treated with alcohol to reach a 5% ethanol concentration and supplemented daily with 10% ethanol. Control flies were not treated. Upon reaching the 3rd instar stage of the Drosophila larvae, experiments were performed. To answer the question how gene expression changes, we performed RNA sequencing, which would allow us to see the mRNA transcripts in the larvae's brain. 3 replicates of approximately 30 brains from each group were dissected, for a total of approximately 90 brains from each group. The results from this sequencing showed 193 downregulated transcripts, that is 193 genes that were reduced in amount, and 258 upregulated transcripts, or genes that increased their amount. Interestingly, many of the enriched genes are highly important to synaptic function and regulation. Additionally, many have been implicated in cognitive processes. Something else worth noting is that after evaluation of some of these genes, we have elucidated many interactions between them, which may give us an idea of what ethanol is primarily affecting in the brain. To answer the question, what other behaviors change, flies' preference to different odors was measured using the preference maze assay. In this assay, adult flies are placed in the assay with a substance entering each channel from one side, here represented with the blue tube. A preference index is then measured. The higher the preference index, the more attraction is seen towards the substance. So, before we move on, it is important to recall that in the next figures, the first 2 graphs compare the preference indexes between experimental, chronic ethanol, and control, ethanol naive, groups of each sex. In the y-axis, we have the preference index, and in the x-axis, the time of the experiment. The third graph shows a summary of results from both sexes and treatments during the first 3 minutes of the experiments. We show these first 3 minutes, given that when arriving at 4 to 5 minutes, this preference is no longer observed as significantly, since the channel is saturated with ethanol. In the first 3 minutes, there is no saturation and the preference is clearly observed. In these figures in particular, we compare attraction to ethanol, where we see experimental females have a lower preference for ethanol, while males do not experience this decrease in preference. This is interesting to us, given that Drosophila females will naturally choose ethanol to lay their eggs. In this particular slide, you can see that the preference for vinegar is compared rather than for ethanol. This time, females do not experience a decrease in attraction to vinegar, but experimental males do. In this case, this is interesting because vinegar is naturally attractive to fruit flies, so it raises a question as to why these flies are less attracted to it. Lastly, we compare attraction to benzaldehyde, a naturally aversive odor to Drosophila. Again, we see a change in preference only in the experimental males but not in the females. In this case, the change seen in males is an increase in attraction. Once more, we are interested given that these chronically exposed flies seem to be more attracted to an aversive odor. To summarize, with RNA-seq, we saw approximately 451 differentially expressed genes in these experimental flies, and those with higher enrichment or larger changes are key to central nervous system synaptic regulation. The preference maze assay allowed us to see evidence and significant sex differences in attraction to different substances. Especially in ethanol, female flies experience less attraction after being grown in ethanol-containing food. Our future directions include the validation of genes seen in the RNA-seq using photogenetics approaches and the larvae heat shock olfactory conditioning assay, which is the assay that was used in Robinson's study. Additionally, we also plan to evaluate synaptic function by electrophysiological analysis and to see what other behaviors might be compromised by chronic ethanol exposure in addition to those evaluated so far. Thank you so much for your attention. I hope you learned something. If you have any questions, suggestions, or comments, please do contact me. Have a great day and enjoy the rest of the conference.